In the previous class of Mendelian genetics, we studied monohybrid inheritance, monohybrid cross, and the results and the law of segregation. So now let us continue with the dihybrid cross. What is dihybrid cross? The cross between two plants differing in two characters is considered as dihybrid cross, or as I already made it clear in the previous class, when you consider the inheritance of two characters together, that can be considered as dihybrid cross. Now Gregor Johann Mendel wanted to know what will be the pattern of inheritance if I consider two characters at a time, right? So he then noted down the inheritance of two characters from parent to offspring. So Mendel considered two characters of pea plant, and in this class we are going to study the example of inheritance of round shaped seeds. So he selected a plant which is pure line, which is producing round shaped seeds with yellow color cotyledon. So now he is considering two characters. One is shape of the seed and color of the cotyledon. So one plant is having round shaped seed with yellow color cotyledon, like this, round shaped with yellow color cotyledon. And uh, another plant which is producing wrinkled seeds with green colored cotyledon, right? So he's going to cross these two plants, round shaped yellow colored seed producing plant and wrinkled seed with green cotyledon color plant, right? And the results he obtained. So let us try to understand what is the result. A pea plant with yellow round seeds is crossed with a plant having green and wrinkled seeds. So that's what I already told you. You can consider the yellow round as the phenotype of one parent and green and wrinkled as the phenotype of another parent. In the F1 generation, all the plants produced yellow and round seeds. Since we already studied in the monohybrid cross and we already understood what is dominant character. So here you can say the character which gets expressed in the F1 generation is the dominant character. It is very easy to decide which character is dominant. When you are crossing two pure line plants and the character that gets expressed in the F1 generation is the dominant character. So here in the F1 generation all the plants were yellow round. Therefore, you can consider yellow color as the dominant character and round shape as the dominant character. Two F1 plants were crossed to obtain F2 generation. So the same thing is done here, and F2 generation plants produced four different types of gametes each. So we'll understand why we have to consider four different type of gametes here. The F2 generation plant showed four type of phenotypes in the ratio of 9:3:3:1. That is the result he obtained. In the F2 generation, he obtained four different types of phenotypes. Remember, there are only two parental phenotypes, but in the F2 generation, he is getting two additional phenotypes. So, what is that? Let us try to understand. With this picture, say this is the parent, capital Y, capital Y, capital R, capital R. Y represents the gene for coloration and R represents the gene for shape. So capital Y represents yellow color and capital R represents round shape, whereas the small y represents green color and small r represents wrinkled shape of the seed. So since these two parents are pure line, are homozygous, so the first parent is having, for each character there are two alleles. And how many characters we are studying now? Two characters. So therefore we have to consider four alleles, right? Two genes are there, so each gene is having two copies. So therefore here we are counting four. So Y, R, R, capital Y, capital Y, capital R is the genotype of this first parent. Small y, small r is the genotype of this second parent. It is going to produce gamete. At the time of gamete formation, one allele of each character must enter into the gamete. So Y, capital Y, capital Y. Out of these two, one will come, and capital R, one from this will come into the gamete, right? So therefore, since only capital Y and only capital R are there, all the gametes produced by this individual will be having capital Y and capital R genes. And the gamete produced by this individual will be carrying only the small y and small r genes. So when these two are crossed, we will get F1 generation. So when these two get fertilized, the F1 generation genotype will be capital Y, capital R, small r. It is heterozygous because one Y is coming from this parent and another Y is coming from here. So here we have capital Y, therefore capital Y, and here we have small y, therefore small y. Capital R, and here small r, small r. So the F1 generation's genotype is capital Y, capital R, small r. Then what is the phenotype? In his experiment, Mendel proved that he obtained all the plants producing yellow colored seeds with round shape, right? Yellow colored cotyledons with round shape. So all the F1 individuals are yellow round, and he crossed them. Now to understand the gametes, we are going to use this square, because like the monohybrid cross, we cannot directly write. Here each plant is going to produce 4 different kinds of gametes. Here, right, this plant, F1 plant, is going to produce a gamete with capital Y, capital R, and another gamete having capital Y and small r, and another gamete with small y, capital R, like this. So each plant will be producing 4 different types of gametes. So F1 into F1 means these 4 gametes into 4 gametes of the same type. Now each gamete is having equal chance of fertilizing the other gamete. So that we have to consider. And to obtain the F2 generation, we have to use the Punnett square, right? So this is the Punnett square or checkerboard we are going to use to understand the genotypes of the offsprings which are produced in the F2 generation. Now let us assume these are the gametes from one parent, F1 generation, and these are the gametes from another generation. So these are the sperms and these are the ova. Here we should not use the word sperm because you can consider them as pollen grains, and here they are of course egg or ova. When this gamete and this gamete is fused, what happens? The individual, the F2 individual, will be having capital Y, capital R. Capital Y, capital Y, capital R, capital R. And when this gamete fuses with the second gamete, what happens? Capital Y, capital R, small r. So therefore you have the genotype capital Y, capital R, small r. And this, when it fuses with the third gamete, it will produce capital Y, capital R, small r. And lastly, capital Y, capital R, small r, like this. So you can go on writing the genotypes when these gametes are produced. So you have to do like this. This gamete fuses with first one, second one, third one, fourth one. You write the genotypes in the first row and come to the second row. So consider the second gamete. So fuses with the first, second, third, fourth. So you get the genotypes of the second row. In this way, we are going to get 16 different combinations. But whether there are 16 different combinations or one less than that, we can understand. You observe here how many genotypes are there. So this is one genotype, capital Y, capital Y, and capital R, capital R. Only one is there, right? And if you observe different genotypes like capital Y, capital R, small r. Here there is one, right? And here another one. So only 2 are there, right? and capital Y, small y, capital R, capital R. So you have them here and here, right? So the number varies. You can observe them later. But we are interested only in phenotypes here. So what is the phenotype of this first plant? Since both dominant genes are present, it will be yellow round. Here also it is yellow round, yellow round, yellow round, because both dominant genes, whether the dominant gene is present in one or two copies, that doesn't make difference, because if only one copy is present, then also it is going to express. Therefore, since capital Y is there, it is yellow, capital R is there, it is round, whether in one copy or two copy. This is also yellow round. But come here, it is yellow because it is having capital Y, but R, you observe the R, both the R genes are recessive here, therefore it becomes wrinkled. This is wrinkled, it is yellow wrinkled, again it is yellow round and it is yellow wrinkled, right? So if you observe the Punnett square or the checkerboard, we identify four different types of phenotypes. One phenotype is yellow round, another phenotype is green wrinkled. We know these two phenotypes because these are the parental phenotypes, right? The yellow round and green wrinkled are the parental phenotypes. But green round and yellow wrinkled are the two new phenotypes which appear in the F2 generation. So green round and yellow wrinkled combination appeared in the F2 generation. It is not there in the parent. So this led to the second law of Mendel, or Mendel's law of independent assortment. So if you understood this and if you count the numbers, we have 9 yellow round plants out of 16. I am telling 3 green round, 3 yellow wrinkled and only 1 green wrinkled. So we have the plants 4 different phenotypes in 9:3:3:1 ratio. So what is the phenotypic ratio in monohybrid cross? 3:1. And what is the phenotypic ratio of dihybrid cross? 9:3:3:1. Remember this. This is universal in all the organisms which follow Mendelian pattern of inheritance. Say when you cross two individuals with two characters, I mean two different expression of the same character, like tall and dwarf, if you get 3:1 in the F1 generation, if you are considering only one character, and if you are considering two characters, you will get 4 phenotypes with 9:3:3:1 ratio. So 3:1 and 9:3:1 3:3:1 ratios are considered as Mendelian pattern of inheritance. So what I am trying to tell you is, if you conduct same experiment in another plant and if you get the same ratio, you must say that the plant is following Mendelian pattern of inheritance. The character follows Mendelian pattern of inheritance, right? If you get the result different from this ratio, then you can say the characters represent deviations from the Mendelian pattern of inheritance. That's what I told you. At the time of Mendel's life, some other scientists, when they conducted experiment on some other plants, they got different results. And Mendel thought he was wrong. No, he was not wrong, because most of the organisms follow Mendelian pattern of inheritance. But there are some characters, there are some organisms and characters which do not follow Mendelian pattern of inheritance. They show different type of pattern of inheritance, and we know why it happens and how it happens now, right? So dihybrid cross shows 9:3:3:1 phenotypic ratio. Based on the dihybrid cross, let us understand the next law of Mendel, that is law of independent assortment. This is important, law of independent assortment. The factors for two or more pairs of contrasting characters are distributed independently of one another at the time of gamete formation. What are the first law? Law of segregation told the gametes produced are pure for a character. And here what the Mendels Law states, the factors for two or more pairs of contrasting characters are distributed independent of one another at the time of gamete formation. I'll tell you how it happens. For example, when Mendel studied the inheritance of factors for rounded and wrinkled shape of seeds and the yellow and green color of cotyledons from the yellow round and green wrinkled parents, he could get some plants like yellow wrinkled and green round. This is due to the independent assortment of factors during gamete formation. Okay, let us try to understand what this law states. See, these two are the parent plants. And what is the genotype of this particular parent? You can say dominant parent, capital R, capital R, capital Y, capital Y. So with capital R, what is here? Capital Y, right? So in this plant, in this parent, capital R and capital Y are together. And you consider another parent, recessive parent. Here, what is the genotype? Small r, small r, small y, small y. Here, small r is present along with small y. What Mendel's law of independent assortment tells us is that there is no affinity between the alleles of different characters. See, in the parent, capital R is present with capital Y. And in another parent, small r is present with small y. That doesn't mean capital R and capital Y have affinity. They will move to the next generation together. No. Since in the parent, small r is present with small y, that doesn't mean they go together. At the time of gamete formation, they will get distributed independent of each other. Now, let me tell you what happens if there is affinity. You think of a classroom where, in each desk, let us say 3 students are sitting. And there is no rule for sitting in the classroom, let us assume, and you will sit with your friends, right? Say the sitting arrangement is random. Anybody can sit in any desk. If it is the condition, what happens? Normally, friends will sit together. In the first desk, 3 students are there and they are normally friends. In the second desk, 3 students, they are close friends. Third desk, close friends. Say one class is over. Now next, you have to move to the next classroom for the next class. How you will move? All the students will move to the next classroom. But here, the students who sat together in the first desk, the tendency is that they will go to the next classroom and there also they will sit together because they have affinity. They are friends. But in nature, that doesn't happen. Say here, you just imagine capital R is sitting with capital Y. So it is going to the next classroom. What is next classroom? Next generation. No, they will get distributed randomly. Capital R may go and sit with small y. Capital Y may go and sit with small r. That's why we got 2 more combinations here, right? So these are the 2 parental combinations. We obtained yellow round in 9 ratio and green wrinkled in 1 ratio, but 3 and 3, 9:3:3:3:1. So 3 and 3. So we obtained yellow wrinkled. So yellow is due to the presence of capital Y, whether homozygous or heterozygous. So here you can see the yellow color seeds are produced and the seeds are wrinkled in shape. That means the capital Y is now present with small r. So there is no affinity between capital R and capital Y, small r and small y. So they get separated independent of each other, right? So this is called law of independent assortment. Now why it is important? In the Mendelian genetics, in the dihybrid pattern of inheritance, we observe the independent assortment, genes moving independent of each other. But later, when many other experiments were conducted, the scientists observed another behavior where some genes have tendency of moving together, right? That we call it as linkage. So when you study linkage topic, you will understand it. But when Gregor Johann Mendel studied the pattern of inheritance, luckily the genes he studied, the character he studied, the genes for those characters were situated in different chromosomes, therefore they moved independent of each other. So you remember, if there is no linkage between the genes, they will move independent of each other. That is the law of independent assortment. Right? So I hope you understood what is dihybrid cross and what exactly is the meaning of law of independent assortment. If you understand this, so then it will be very helpful for you to understand the linkage topic in the later classes. Okay, that's about the monohybrid and dihybrid cross. In the first class, we understood monohybrid cross, and in this class, we understood the dihybrid cross and law of independent assortment. Okay, then, thank you.